Welcome to the Advanced Fluorescence Microscopy and Image Processing course. So today is the fourth lecture, that is the second lecture of second week, and today we will learn about fluorescent proteins, organic dyes, and the labeling strategies. So in many research articles or in several presentations, you must have seen this kind of beautiful images of the cellular structures and cell organelles, in which different cellular organelles or the cellular proteins, they are labeled in different colors, and you can merge all those colors to see their co-localization. So how today in today's class we'll learn how this kind of different proteins or the cell organelles can be stained with different fluorescent colors, and how we can visualize them under the fluorescence microscope. So in this image you can see this is the merged image of all colors, but individual colors shows different structures, like chlorophyll are shown in blue color, then mitotracker shows the mitochondria which are in yellow color, then some other protein is tagged with GFP, and other protein is tagged with the dye known as FM464 and by Venus color. Okay, so different proteins or the cell organelles can be labeled in different colors, and you can merge all the colors to see their relative localization inside the cell. So today we'll learn how to do this. So there are several protein labeling strategies. The first one, which is very common, is known as the genetic encoding. And so this genetic encoding takes place at the C terminal or the N terminal of the gene of interest. So let's say you are interested in visualizing the intracellular location of protein X. So what you have to do is you have to fuse this gene X, which codes for the protein X, with the at its C terminus with the GFP. So GFP is the green fluorescent protein, the gene for the gene fluorescent protein. So this kind of genetic encoding is taken place at the endogenous level. So in the genome of any organism, you have to fuse this GFP gene at the C or N terminus of your gene of interest. So there can be two types of fusion. Either you can fuse this GFP gene at the C terminus of your gene of interest, or you can fuse it at the N terminus of your gene of interest. So there are several advantages and disadvantages of this approach. So what are the advantages of labeling any protein by this endogenous protein fusion? So the advantage is that it offers specific labeling, because you are endogenously putting this GFP tag at the C or the N terminal of your gene of interest. So only your protein of interest will be GFP tagged. There is no chance that any other protein will give you the green colored fluorescence. So it is known as the specificity of labeling. The second thing is it is essential for live cell imaging. So if you want to plan an experiment in live cells, not in the fixed cells, then you have to go for this option. If you want to study some protein dynamics and all, you have to tag your protein with a fluorescent protein. Okay. And the disadvantage is that the time required for genetic engineering is generally too long. So generally, this kind of endogenous protein fusion or the gene fusion takes 3 to 6 months in the laboratory condition to generate this kind of strains or the constructs, because it requires extensive amount of cloning and some transformation or transfections in the cell lines. So that process takes generally 3 to 6 months in the laboratories. So there is another option which is known as the antibody-based labeling or the antibody-based detection, which is known as immunofluorescence or the immunohistochemistry. So this is quite rapid approach which doesn't need 3 to 6 months of time. You just buy the protein specific antibodies commercially, and you can label your protein of interest with the specific antibodies. So you can see here in the image, let's say this is your antigen. That means the protein of your interest which you want to visualize inside the cell. So it has one specific binding sites for the primary antibody. So primary antibody is specific for this protein, and that primary antibody binds to this protein. And at the FC region of the primary antibody, you can see the fluorophore, that is a fluorescent dye attached to the antibody. So wherever this antibody will bind in the cell, you will get some fluorescence. Now there are two ways of doing this immunofluorescence. So ideal means either you can have the primary antibody itself labeled with the fluorophore, or sometimes people use the primary antibody without any fluorophore, but they use the secondary antibody which is specific for the primary antibody, and that secondary antibody is labeled with the fluorophore. So this kind of labeling strategy is known as indirect immunofluorescence, where you actually use a secondary antibody which is fluorescent uh, that binds to the primary antibody. And this kind of immunofluorescence is known as the direct immunofluorescence, in which the primary antibody itself is labeled fluorescently. So again, there are several pros and cons of this approach as well. So the pros are that there is no genetic engineering is required. So the time requirement is less, because most of the antibodies are available commercially these days. So you can just buy those antibodies, though they are very expensive, but they can save your time. Second thing is, this approach is suitable for only fixed cells, because there are several agents, fixation agents used for this immunofluorescence. So cells, you cannot label the protein of your interest in the live cells with this approach. So for seeing the visualization or localization of the protein in the fixed cells, this approach works well. And another disadvantage is some level of non-specific binding may occur if the primary antibody that you are using or the secondary antibody that you are using, it has some non-specific binding affinity. So sometimes you may see some non-specific binding, but there are ways to reduce the non-specific bindings. The another approach, which is quite recent, that is used for labeling the proteins in cells, which is known as a covalent modification. So there are several methods for that. The first method is known as self-labeling enzyme derivatives, or the tags. So there are some tags available which are actually small protein moiety, which are known as halo tag or snap tag or clip tag. So the way you fuse your gene with GFP or the green fluorescent protein, similarly you have to fuse your gene at the endogenous locus with a halo tag or the snap tag or the clip tag. And there are fluorescent ligands which bind specifically to halo tag or snap tag or clip tag. So those fluorescent ligands are available commercially. You have to buy those dyes, and those dyes you have to add into the medium. So those fluorescent dyes will go inside the cell and bind to this halo tag protein or the snap tag protein or the clip tag protein. There are several other ways known as chemical labeling and enzymatic labeling. So by several chemical modifications and the enzymatic reactions also, you can label your protein of interest with fluorescent dyes. So now let us understand a little more about the discovery of the green fluorescent protein. So the discovery of green fluorescent protein, which is known as GFP, leads to revolution in fluorescence microscopy. So the prospects of fluorescence microscopy changed dramatically with the discovery of the fluorescent proteins in 1950s. So initially in 1950s, this green fluorescent protein was first discovered in the jellyfish Aquaria victoria, and Nobel Prize was awarded to three scientists, Roger Shen, Osama Shimomura, and Martin Shelfie in 2008 for the discovery of GFP. So if you see this jellyfish under the seawater, it emits green colored light. So you can see some kind of a light emitting creatures in the seawater. So that is a jellyfish. Now why this jellyfish emits green color light? So that emits green color light because of the presence of a specific protein which is known as green fluorescent protein. And if you see the three-dimensional structure of GFP, it looks like this. So because of some specific arrangement of the amino acids, three amino acids at specific positions, such as serine at 65, threonine at 66, and glycine at 67, this protein gives fluorescence when it absorbs blue light. Okay, so the major excitation peak of the original GFP is 395 nanometer. The minor excitation peak was at 475 nanometer, and the emission peak is at 509 nanometer. And the molecular weight of this GFP protein is around 27 kilodalton, and it is made up of 238 amino acids. So if you fuse this GFP to any of your protein of interest, you can see inside the live cell, wherever your protein will go, the GFP will follow. So here you can see some specific labeling of some intracellular structures or the proteins by the use of GFP. Even in the entire animals such as mice, you can fluorescently label some proteins, and when you illuminate them under the UV light or 488 with 488 nanometer, you can see the entire mouse gives green color fluorescence. So this discovery of green fluorescent proteins made this possible to visualize the intracellular structures and dynamics using fluorescence microscopy. Now, there were some disadvantages of this original GFP, which was discovered from jellyfish Aquaria victoria. So the first disadvantage is it is pH sensitive and it has tendency to dimerize. So this tendency to dimerize is not quite a beneficial feature for any kind of biological activity, because you label your protein of interest with GFP, you fuse your protein of interest with gene of interest with GFP, and whatever the protein will produce, it will start making dimers, and that may hamper the biological activity of that protein. So how to avoid this kind of problems? So to avoid this kind of problems, scientists started making several genetic manipulations to change the spectral and physical properties of GFP. So see which kind of genetic engineering was done. So to reduce the dimerization, scientists replaced the non-polar amino acids with hydrophilic amino acids. So these are the conversion of 3 amino acids that they did in the original sequence of GFP. Converted F amino acids present at the 223 position to R. Now which one is the F amino acid and the R amino acid? So I have given the amino acid codon table here. So from here you can find out, so F represents phenylalanine, and they converted phenylalanine into R. So R stands for arginine. Okay, so they converted phenylalanine into arginine at 223 position. Similarly, L at the 221 position to K. So L stands for leucine, and they converted it into lysine at this position, and A206 K. So A stands for alanine. So they converted alanine to lysine at the position 206. So see, this protein is made up of 238 amino acids. So these are the positions of amino acids at this different locations: 223, 221, and 206. So by just changing some of the amino acids, you can change the spectral and physical property of the GFP, and this process is generally known as genetic engineering or the protein engineering. Similarly, several more genetic manipulations were carried out to change the spectral and physical properties of GFP, and see what happened. So when they converted S65 to T, it gave rise to the new variant of the GFP, which is known as enhanced GFP or eGFP, and this eGFP is 5 times brighter than the original GFP. Then they did this conversion at T203. They converted T at 203 position to I, and that leads to alteration of the excitation maximum to 399 and emission maximum to 511 nanometer. So it changed the excitation and emission spectra, and it changed the color also. So from GFP, now it is a sapphire color. Then by changing this Y66 to H, the color changed to blue. So that is known as the blue fluorescent protein. Then changing Y66 to W, it changed the color to cyan. So this fluorescent protein is known as cyan fluorescent protein. Similarly, this by changing just 1 or 2 amino acids, they converted into different colors like YFP, yellow fluorescent protein, and eventually GFP and its variants covered the entire wavelength spectra of the visible light. So you know that this is the wavelength spectra of the visible light. So by modifying certain amino acids, they could develop all these different proteins in this range, from blue fluorescent protein, cyan fluorescent protein, yellow fluorescent protein, green fluorescent protein, okay, and sapphire fluorescent protein. Now what is missing? The red color fluorescent proteins are missing. Okay, so this spectrum of the visible light, we do not have any fluorescent protein to use in the life cells. Okay, so what happened? So before going into that, let me explain that here you can see colors of GFP mutants expressed in E. coli. So the E. coli are the bacterial cells. They are plated on this media or on the Petri plate, and you can see they emit different colored light because they contain different proteins like YFP, BFP, CFP, and GFP. Now, how the red color fluorescent proteins were developed? So, to fill this gap of red fluorescent proteins, a Russian scientist Sergei Kukyanov discovered the red fluorescent protein in Anthozoa. So, red fluorescent protein have a big advantage over others because there is much less autofluorescence in the cells in the red spectrum, and they are excited by a longer wavelength, which is good for live cells because it creates less phototoxicity. The red wavelength creates less phototoxicity compared to the green wavelength. So anthozoa proteins mature well at 37, and they do not need any genetic manipulations like the original GFP. So several variants of this red fluorescent protein were developed, which is known as DS red, so with the excitation of 558 nanometer and emission of 583 nanometer. And the disadvantage of this DS red version was longer maturation time and tetramerization. So it tetramerizes. Okay. So the further modifications were made by changing or mutating some of the amino acids. So the second version came, that is known as DS red 2, which matures faster and reduced oligomerization. And then the further advancements developed mRFP1. So that is the first monomeric RFP. So see, the original red fluorescent protein and the DS red, they tend to tetramerize. Okay, but this was the first monomeric red fluorescent protein. And further genetic manipulations to mRFP1 leads to 6 M fruit fluorescent proteins known as M honeydew, M banana, M orange, M tangerine, M strawberry, and M cherry. Okay, so based on the colors of the fruits, they were named with that fruit name. So M cherry is widely used, which has emission of 610 nanometer. It has the brightness 50% of the EGFP. M plum emission is 649 nanometer, and MCAT2 has the emission of 635 nanometer. So MCAT2 is the brightest red fluorescent protein, whereas M plum is the red fluorescent protein with highest emission wavelength. And here you can see on this slide, you can see these are the currently available fluorescent proteins with different excitation and emission wavelength. And you can see that now we have entire series of fluorescent proteins in the entire visible spectrum covering all the wavelength. So you just have to select the best colors based on your requirement. And see, they are named, some of them are named with the name of the fruits, like this is a enhanced blue fluorescent protein, enhanced cyan fluorescent protein, enhanced green fluorescent protein. Then this is YFP, yellow fluorescent protein. This is M honeydew, M banana, M orange, TD tomato, M tangerine, M strawberry, M cherry, M grape 1, M raspberry, M grape 2, M plum. So based on the colors of the fruits, they are named differently. Now, how to select the best fluorescent protein for your whatever the application is. Okay, so here I have given the link to the fluorescent protein database. Okay, so before choosing any fluorescent protein, just visit this link. I will just open it and show you. So this is the fluorescent protein database FPbase website, and here any fluorescent protein if you want to use, let's say you want to use M cherry. So write M cherry, and it will show you all variants of M cherry which are currently available. Okay, and let's suppose if I'm selecting this M cherry 2, so it will provide all information about this protein. This protein has unmoderate changes. Click here to view the last moderated version of this protein from August 10 or view the full change history. Okay, so see what other information this database provides. It shows you the entire history of this protein. So see, originally it was DS red. It was published in 1999. Then some genetic manipulations were done to produce this DS red express in 2002. Then further genetic manipulations were done to produce this version, which is dimer 1 version, in 2002 only. Then the mRFP1 was developed in 2002. Then mRFP 1.1 was developed by further genetic manipulation of this version in 2004. Okay, and then in 2004 this version was developed, and in 2004 another this version was also developed by some other group. Okay, And this website also provides you the excitation and emission spectra of any fluorescent protein, and it gives you some additional useful information like oligomerization. So this is the monomeric protein. So this is good for your labeling, because it will not oligomerize or tetramerize or dimerize. Okay, so generally you should use the fluorescent protein which are generally available in monomers. And its molecular weight is 26.7 kilodalton. And these are some other biophysical parameters such as excitation wavelength, emission wavelength, uh, its maturation time. So once this protein is produced, it takes 22.8 minutes to give the fluorescence. So the maturation time is quite high for this protein. And about some more details about its photostability, its sequence. If you want the amino acid sequence of this protein, you can get it from here. And you can also get some advice from the users of this protein. So before using any fluorescent protein, just check this database and get the enough information about that fluorescent protein from this website. Okay, let us go back to the slides. These are all available fluorescent proteins in different colors, and now we'll see some more advanced versions of the fluorescent proteins. So these fluorescent proteins, such as what are the colors that we saw in the previous slide, and GFP, YFP, DS-RAD, are powerful tools to visualize the cellular components. However, they cannot provide the dynamic information about protein turnover and spatiotemporal localization. So to study the protein dynamics, these new versions of fluorescent proteins were developed, which are known as photoactivatable fluorescent proteins, photoconvertible fluorescent proteins, and photoswitchable fluorescent proteins. So what is photoactivation? So photoactivation means initially that fluorescent protein remains dark, but then you have to use UV or the blue light to activate it, and then only it gives you green fluorescence. So this is known as photoactivatable GFP. The original GFP is here. This is the eGFP, enhanced GFP. From the beginning of your imaging experiment, it will show you green fluorescence. But if you use this version of the GFP, that is photoactivatable GFP, so initially you will not see any fluorescence. But then you have to induce the fluorescence by using the blue light. Then that fluorescent protein will start giving the fluorescence. The other types of proteins are known as photoconvertible fluorescent proteins. So what is photoconversion? So initially the protein is green in color, but if you use UV or blue light, it turns red. And in the next slides, I will show you how these properties are useful in studying the protein dynamics in biological systems. And the photoswitchable fluorescent protein. So they can convert from red to green and green to red by using different wavelength of light. So if you use the 405 nanometer wavelength, you can convert the red protein into green. And if you use this 503 nanometer light, you can convert this green protein into red. And there are here some of the names of these proteins are given, like Dronpa, Dreklang, kindling fluorescent proteins. So these are the names of this photoswitchable fluorescent protein. Then Dendra2, CAD, EOS fluorescent proteins are the types of photoconvertible fluorescent proteins. And PAGFP, PM-MCherry, PA-MCAT, they are photoactivatable fluorescent proteins. So let us see how these three different versions of these fluorescent proteins are useful for studying protein dynamics. So here you can see an example of a photoactivatable fluorescent protein. So see, initially in the cell there is no fluorescence, but then using the blue or the UV light, we can induce the fluorescence, let's say in the nucleus. So only the fluorescent protein which is present in the nucleus, it turns on and it gives green fluorescence. And then you can image this cell for longer time to see whether this protein is localized only in the nucleus or it comes out from the nucleus as well. So if this protein is dynamic, if it is coming out from the nucleus, you will see that this green fluorescence you will gradually see in the entire cytoplasm also. But let's say if this protein is stable, it is just let's say chromatin bound, it remains only inside the nucleus, then you will see that after some time you will not see the fluorescence in the cytoplasm. But this protein is highly dynamic, so that after few frames you can see that this fluorescence which you activated only in the nucleus, but now it has come to the cytoplasm. So this kind of fluorescent protein, before activation, you can see just the dark background, right? But after giving this blue light, you can induce the fluorescence, and the fluorescent protein gets activated. It gives you the green fluorescence. So this is known as photoactivatable fluorescent protein, because initially the fluorescence was not there, but then you have to activate it to see the green fluorescence. Then coming to the photoconvertible fluorescent protein. So what is photoconversion? So here you can see initially this entire cell is filled with the green fluorescent protein, but then with the help of blue color or the UV light, you can convert the protein molecules which are present in the nucleus into red. And then you can see the cell for some longer time, and you can see that whether red protein is the molecules which are in red color, they are coming out from the nucleus. So eventually after few frames you will see both fluorescence, red and green. So eventually it will look like yellow in color if you merge both the channel, because the red protein which was present inside the nucleus, it has come out, and the green protein which was outside the nucleus in the cytoplasm, it has gone into the nucleus. And that's why the entire cells gives you both red and green color fluorescence, and if you merge them you can see the yellow colored fluorescence. Okay, here you can see the real example of this photoconvertible fluorescent protein. So see, this structure is stained, whatever this protein or the microtubules or whatever it is, or the actin cytoskeleton, it is stained in red color with a red fluorescent protein. And then they make this specific ROIs, region of interest, to convert those areas into green by the UV light. So in the microscope, especially the confocal microscope that you will see, you can pinpoint such kind of small areas for illumination by the lasers. So the blue light laser will illuminate only these three region of interest. It will convert the protein from red to green, and then you will see whether this green protein remains in the same area or it travel to the other locations. And after some time you can see that this green protein which you converted only at this area, but now it has diffused through all other areas in the cell. That indicates that these protein molecules are not actually stable molecules, but they are dynamically exchanging with their surroundings. Whereas here you can see that after ethanol treatment, if you bleach these three area, then there is no exchange of these molecules, because now the cells are fixed. So they will not, all the protein movements are stabilized. So now you will not see any exchange of the green fluorescent protein into the red area and the red fluorescent protein into the green area. So this is the application of the photoconvertible fluorescent protein. Here another example is there. So this is pre-photoconversion. So you can see whatever the structure is in the green color, but then they make a small region of interest to uh, illuminate it with the blue light. So they converted this area into red. So you can see that now the green fluorescent is absent from this area and the red fluorescent turned on, because what are the fluorescent protein molecules were that they are converted in the red into red color. And then after some time you can see that those molecules spread around and they can make this kind of grid like structures. And so basically it shows that these protein molecules are not stable, they're actually diffusing, they are actually exchanging with their surrounding molecules. So this way you can study protein dynamics using photoconvertible fluorescent proteins. So here you can see an example of the photoswitchable fluorescent proteins. So you can see initially the entire structure is illuminated and you can see the green fluorescent protein everywhere. But then they can switch off the molecules which are here, and only some specific structures here they are shown by FVIO. Okay, I don't know what is the full form, but whatever is the full form, you can selectively select the region of interest and switch off the fluorescence from the other part of other areas, and again you can switch on that fluorescence and switch off this fluorescence using a different color light. So this is known as photoswitchable fluorescent proteins. So they are also used for studying protein dynamics. By now we discuss only about the first type of protein labeling strategies, which is known as the genetic encoding, because for all this fluorescent protein you have to genetically fuse them to the C terminus or the N terminus of your gene of interest, whether it is a photoactivatable or convertible or photoswitchable fluorescent protein, or it's a regular fluorescent protein. You have to do the C or N terminal fusion with your gene of interest. So now I will show you some of the fluorescent dyes which are used for this antibody based detection in the process known as immunofluorescence or immunohistochemistry. So what are fluorescent dyes, how they are different from the fluorescent proteins, and what are the advantages that are being offered by the fluorescent dyes? So fluorescent proteins can only be used when the genetic engineering is possible with the cell. However, there are certain instances when genetic engineering is not possible, such as histological samples. Immunofluorescence staining are used to visualize the protein of interest. So as I mentioned that for using the fluorescent protein, you have to go for the genetic engineering, which is a time-consuming process, which generally takes 3 to 6 months in the laboratory condition to generate the fusion proteins. Okay. But what, let's say if you are finding some cells from the cancer patients, and if you see that whether some particular protein is localized properly or not. So you cannot wait for 3 or 6 months for those kind of experiments, right? So for that purpose, you will take help of the immunofluorescence, in which you will buy the commercially available antibodies for your protein of interest, and you can perform immunofluorescence, and you can use these dyes for the fluorescent labeling. So here are some of the disadvantages of fluorescent proteins over fluorescent dyes. This Now let me explain first what are fluorescent dyes. So see, as we saw in this slide, there are so many colors of fluorescent proteins are available, and these fluorescent proteins are used for the live cell imaging applications only. Okay, but fluorescent dyes are also available in so many colors, and they can be used for fixed cells. So here you can see this is the entire spectra of fluorescent dyes. They are known as coumarins, rhodamines, and cyanine dyes. Okay, they are available in entire visible spectrum. So you just have to select the color of your interest to label your protein of interest. Okay, and so these dyes are available in so many different colors. And what advantages it offers? So use of fluorescent protein requires genetic engineering, which is a time-consuming process, whereas using the fluorescent dye for imaging is not that time-consuming. Then fusion of the protein of interest with the fluorescent protein increases its molecular weight by 27 kilodalton, because the molecular weight of GFP itself is 27 kilodalton. So when you fuse it with your protein of interest, the overall molecular weight of your this fusion protein will increase by 27 kilodalton. So that may affect the protein function. Whereas these fluorescent dyes are molecular weight wise, they are very small molecules and not that heavy like a GFP fluorescent proteins. Okay. Also, the fluorescent proteins are photobleached faster compared to the fluorescent dyes. So fluorescent dyes are more photostable than the fluorescent proteins. Fluorescent dyes are brighter than the fluorescent proteins. So fluorescence proteins are can give weak fluorescence, but if you use the fluorescent dyes, you can see the bright fluorescence. And most fluorescent proteins tend to oligomerize. As we saw that most of the fluorescent proteins, they were either dimerizing or tetramerizing, which is not a good feature for imaging in biological samples. So you need to avoid the oligomerization. So these fluorescent dyes do not oligomerize. So they provide this kind of advantages over the fluorescent proteins. And these dyes are nothing but they are just kind of a chemical structure. Here I have shown the chemical structure of one of the dye, which is known as fluorescein, which is equivalent to green fluorescent protein in terms of its excitation and emission spectra. Okay, it excites at the 488 nanometer and it emits at 510 nanometer, exactly like GFP. But it is brighter, photostable, and low in molecular weight compared to GFP. Also, there are quantum dots and inorganic nanoparticles which are also used for fluorescence imaging applications. So quantum dots are the semiconductor particle a few nanometer in size, having optical and electrical properties that differ from larger particles due to its quantum mechanics. Okay, and they are extremely bright and they do not bleach. So let me explain you what are quantum dots, how they are made up of. So see, quantum dots are made up of some kind of a semiconductor metal, such as CDSE or CDTE. Okay, and they are for labeling any biological samples. They have some biological conjugates such as drug, protein, or antibody, and by which they can be conjugated to the proteins or any other biological molecules. So they are basically the metal particle, semiconductor metal particle, and they are available in different size like 2 nanometer, 3 nanometer, 4 nanometer, 5 nanometer, 6 nanometer. And depending on their size, their color changes, that fluorescent wavelength changes. So the smallest molecule, that is 2 nanometer, it emits blue color light. Then the largest one is the 6 nanometer, which emits red color light. So their quantum dots are available, also available in different colors, okay, and in different sizes. But they are not uh, uh, I would say they are not frequently used in imaging biological samples because of certain disadvantages. So see, what are the disadvantages? They are not cell permeable, okay, so they cannot go inside the cell. You need some special mechanism to bring them inside the cell, and they are cytotoxic if untreated. So they are also toxic for the cells. So if you use them for live cell imaging kind of applications, it may uh, uh, create some cytotoxicity. Okay, so that's why the usage of these quantum dots is very less in imaging biological samples. Then there are several non-covalent probes are available commercially with high affinity for biomolecules. So let's say there is a dye known as DAPI. The full form of DAPI is 4,6-diamidino 2-phenyl indole. Okay, so it's a fluorescent stain that binds strongly to adenine thymine rich regions in DNA. It is extensively used in fluorescence microscopy to stain the nucleus. So in most of the microscopy images, you will see that the nucleus is stained in blue color with a dye known as DAPI. Okay, so you just add that DAPI to the cell culture or on the fixed cells, and it will bind to your AT-rich region in the DNA, and it gives blue color fluorescence. So this dye is used as a marker for nucleus or marker for DNA. Then phalloidine is another dye. It is a toxin that binds to F actin, and hence to visualize actin cytoskeleton, fluorescent label phalloidine is used. So let's say if you want to label the actin cytoskeleton in the cell, phalloidine is used. So though phalloidine is toxic for the cell, but you can use it on the fixed cells, and phalloidine emits red color light. So here you can see the phalloidine staining of the cell. Similarly, several other compartment and organelle specific dyes are available commercially. Let's say mitotracker. So mitotracker is used to stain mitochondria, and this is cell permeable dye with which is available in different colors like green, red, and orange. Okay, so you have to decide the color, that whether you want to see mitochondria in green color or red color or orange color, and use that specific mitotracker dye, and you can label your mitochondria in any of this color. Similarly, the lysotracker dye is used to stain lysosomes. ER tracker dyes used to stain the endoplasmic reticulum. There are some dyes available for labeling Golgi apparatus, lipid rafts, cytosol, and even to label some ions like sodium, calcium, chloride, and magnesium ions can be tracked with the help of fluorescently labeled chelators that change their spectral properties when bound to the appropriate ions. So let's say for calcium detection, this Fura 2 or Indo 1, Flow 3 or Flow 4 and calcium group, these are different dyes available which binds to the calcium ion, and you can actually see the flux or the dynamics of the calcium uptake in the cell. So here you can see a merged image of the cell in which different cell organelles or the proteins are labeled in different colors. So chlorophyll are labeled in blue with a blue color dye. Then mitotracker, the mitochondria are labeled with this yellow color dye. 2 or 3 proteins are labeled with different color dyes like GFP, Venus, and FM464 dye. And if you merge all these colors, you can see the multicolor image like this, which shows you the relative localization of different cell organelles, the chlorophyll, and different proteins where they are localized. So generally in fluorescence microscopy, we can do multiplexing for mostly up to 4 colors. Okay, so you can see 4 things simultaneously because of the limited wavelengths available in the visible spectrum and limited dyes. Okay, you can use 4 different dyes to label 4 different structures or the proteins. Okay, so blue color generally stains the DNA. The dye that is generally used for labeling DNA or the nucleus is known as the DAPI. Then this FITC provides green colored light. So this FITC dye can be used to label any protein. This TRITC is a red colored dye. It is used to label some other protein. And this far red color, that is magenta, is also used for labeling some other protein or the cellular structures. So mainly this is known as the multiplexing. So four different colors you can image simultaneously from the same sample, and you can study their relative localization. So this is known as the multiplexing. When you image more than one color in the same cell, that is known as multiplexing. So I think with this, we'll stop here for today's class. And the next class will be on 6-dimensional imaging, live cell imaging, and time-lapse imaging. Thank you.